Hey guys, so we're looking at section 15.3 here, which is called Studying the Human Genome. And so now we're going to kind of step into how we create these things called genetically modified organisms. But also in doing so, we're going to talk about how we actually research genetic information, what we know about the human genome specifically, and how we go about trying to identify, let's say, the function of a gene and whether or not it gets transcribed and translated into something like a protein. And so what you're looking at here is something called a DNA a gel electrophoresis. I was going to say microarray, but that's not what this appears to be. So gel electrophoresis is something we're going to talk about that really is going to be responsible for separating DNA fragments according to their sizes. And that's going to be something, again, we're going to talk about here as one of the tools, one of the techniques that we use in helping to understand our genome and the letters that make it up, the genes that we find. Okay, so go ahead and get the title, section 15.3, Studying the Human Genome. And with this, we've only got two objectives. So we're going to look at how scientists can read DNA-based sequences, how we identify the sequences of a specific gene and maybe compare those to other individuals. And then we're going to talk about, again, the size of the human genome, what we know about the genes that we carry, and then also what we're still kind of trying to figure out and learn. Okay. So when we talk about DNA information and how we kind of deal with the information that we've got, and I'm going to move myself all the way up there, deal with the information that we've got and really try to understand what it is that makes us humans. We have several tools that we need to use. Okay, so scientists use several different types of processes and procedures to identify what genes do, to identify the sequence of a gene, to alter genetic information in a way that's meaningful, maybe to knock out a gene to see how that impacts potentially a disorder, to manipulate base sequences in a way that may kind of mimic a mutation or assess the impact of that on a protein. We can also use these manipulation tools to kind of modify, recreate genetic sequences in a way that allows us to attach things like luminescent and fluorescent tags to DNA and then to the proteins that are made from that DNA, so we can see how and when genes are expressed. There are a lot of different kind of applications for this. Most of it, I should say, is not controversial, but some of it can be, and we'll get to that more controversial stuff as we get back into 16.2 and 16.3. But in general, they kind of fall into four different categories. These are going to be really, I'm going to talk about just a couple of these today, and then the rest will come in 16.2. So the first one we can see here is cutting. So if we understand where our gene is, and sometimes we do, if we go by that kind of knowledge, then we can identify types of molecules that we know can help us cut or excise and remove our gene of interest from a segment of DNA. And those enzymes are going to be helpful here in that process. We then can separate that gene sequence here through this technique that we're seeing here, which is actually the gel electrophoresis you saw on the first slide. We can copy the DNA sequence that's been cut out of the DNA. We can also assemble it into different orders, into different fragments, and kind of organize that genetic information as needed with this process over here. And I'm kind of alluding to some of these, but not actually using their words because I know they're going to come up here as we go through the remainder, okay? So if we're going to work with a gene in a lab, and let's say that we know what the DNA sequence is, okay, it's been identified, but let's say researchers just simply don't know what it actually does. They don't know if it codes for protein. They don't know if it may code for a disorder. They don't know if it may be mutated. And so when we want to kind of start initially assessing what a gene does, the first thing we want to do is take it out of the chromosome, isolate it, and start to work with it kind of on a one-on-one basis. And so the tool that we use to help isolate a gene is called a restriction enzyme. Restriction enzymes, I always think of as these little tiny molecular scissors. They come through and they chop DNA at specific positions, what we call recognition sequences. So here's a DNA molecule, and we assume, like if this is a eukaryotic chromosome, right, it's linear, so it would have a start and a stop somewhere, but it would be very, very long, right? Well, what these restriction enzymes do is they're going to come in, they're going to bind to the DNA, and they're going to chop up the DNA at any sequence that reads this. So it has on the top CTTAAG, and at the bottom, this is actually called a palindrome because it's repeating the same. It's just kind of a mirror image. GAATTC. And if we read the top again backwards, GAATTC. So this palindromic sequence is where this restriction enzyme, this particular one, will actually cut. And when it cuts always between the G and the A. So you can see that on the top, on the bottom, and over here on the top and on the bottom. So in isolating a gene, if we know the sequence, we can look at what's around that sequence and say, okay, if I want to cut out my gene, I can cut it here and I can cut it here without actually impacting the gene. And then I can take my gene and I can move it over here, right, and do something else with it. So these little restriction enzymes chop the DNA like we're seeing here. And then if this is my gene of interest, I can isolate that and then take it and move it somewhere else or assess maybe what it does through some other type of technique. Okay. But again, restriction enzymes are kind of the first step. They cut DNA. Okay. Now, I mentioned that we want to be able to separate our gene of interest. So let's say I've got my gene, I've cut it with restriction enzymes, but it's in a little tiny test tube and it's just floating in a solution. And, you know, like everything, if we think about DNA, it's teeny tiny. There's no way I can like see it and like separate it out, right? So we have to use a different tool to try and separate our gene of interest and remove all that excess DNA that we're not interested in looking at, right? We just want our one gene. So the tool that we use for this is called gel electrophoresis. And unfortunately, there's not really a shortened term for this one. Oftentimes in a lab, we'll just call them gels. Like you're going to go run a gel. When you run a gel, what you're essentially doing is you're taking your DNA sample, you're going to put it into this matrix that is very gel-like. Like it's flexible. It's like Jell-O in a way. Okay. It can break, certainly. But what's helpful about it is it's not a super solid type of material in a way. So there's a lot of little tiny fibers in there and things that provide kind of like a matrix. And that matrix allows some molecules through, but others not so much. So the shortest molecules tend to be able to travel the farthest because they can kind of squeeze through all those little spaces between the fibers that make up that matrix. But the largest DNA molecules tend to hover kind of and stay up at the top of the gel. So if you take your gel, let's say here's my DNA sample, I'm going to put that in this little well right here. You can see the DNA is floating in that space. And then I take it and I expose it to an electrical current. So this is sitting in a little bath of water. I plug in my current here. I'm going to plug it in and I'm going to run a current from negative to positive. And DNA I know is negatively charged. Which if you think about what that means it naturally wants to migrate this way. It naturally wants to go to the positive charge. And as it moves to that positive charge, again, the shortest fragments are going to move fastest. The longest are going to stay up here towards the top. And so we can essentially see, based on the movement of these, what size these different pieces of DNA are. And so let's say my gene of interest was really, really small, like the smallest fragment that I could have possibly created. It would be this guy way down here. Okay. If it was really, really long, right, it could be any of these three up here. And then everything else is kind of in between. So what you would do from this point, let's say I know that it's the smallest fragment, I can actually physically take my gel out of the little water bath, turn off the current, of course, and then I'm going to use a little scalpel to actually cut out my gene of interest, throw it in another test tube, reliquefy it, and then I've got my gene and only my gene, and I can do all sorts of different things with it. Okay, so from gel electrophoresis, we can then use DNA for fingerprinting, for cloning, for gene sequencing, for polymerase chain reaction, which we'll talk about later, for all sorts of different things. Okay. So one of those things that I just mentioned is called DNA fingerprinting, and this is what it sounds like. Every person, every individual in existence, right, and that will be in existence, has a unique DNA fingerprint based on the genetic information you have in your cells. Well, DNA fingerprinting is kind of like fingerprinting at a crime scene in a way, right? Everybody's supposed to have a unique fingerprint. The difference is this one just doesn't, like, you can't just, I don't know, get rid of it, right? I mean, a DNA fingerprint is going to be present in any type of leftover tissue. So it could be a hair sample, it could be saliva, it could be semen, it could be sweat, it could be little flakes of skin, right? It's present everywhere. And so as long as you've got cells involved, you have access to an individual's DNA fingerprint. And what they essentially do, what forensic scientists would do in this case, is if they had a DNA sample, they would take that DNA sample and expose it to restriction enzymes, and then run that DNA sample out on a gel to see what these little, what we call banding patterns would look like, right? The little lines that are created here, the DNA fragments. Now, if they also had DNA from a crime scene, and a suspect, then they could run them out together. And you could see here in this image, suspect 2, the DNA was clearly at crime scene 1, right? Those bands are perfectly matched. And at crime scene 1, none of those bands really match anybody else. The victim here was clearly found at crime scene 2, or at least the DNA was there at crime scene 2. And suspect 1 doesn't appear to be anywhere because none of their bands matches with any of the crime scenes, nor does it match with, victim or other suspects. So a DNA fingerprint has actually been used many, many times in order to exonerate like wrongfully convicted people for crimes that they didn't commit by analysis of DNA samples, right? They can also be used to convict people that of crimes where they've left DNA behind, right? So it can be used for all sorts of different things. We can also use it to identify the parents of a child, the relatives of someone killed in a war if there aren't a lot of remains left, but there are DNA pieces, I guess. That's a terrible thing to say, but there are, remnants of DNA that are accessible, then yeah, you can use this instead of, teeth and that kind of stuff that we've used in the past. So fingerprinting is a super cool use for that type of technology. In addition to that, gene sequencing. So one of the other things we can do is, and this is one means by which we've been using biotech to help map out the human genome, and I'll talk about that in a second. But gene sequencing is essentially identifying the order of letters. So let's say, again, going back to my original kind of story here, let's say I have a gene that I'm interested in. I don't know what it does. I've taken it out of its chromosome by using restriction enzyme. I've isolated it by using gel electrophoresis. And now I want to make sure that the fragment I actually cut out of my gel is actually the right one. Because sometimes there are fragments that appear to be very similar in size, and it's hard to assess which one is actually your gene, right? It's not always as cut and dry as this, right? The bands are going to be much more closer together, and there's going to be many, many more. Like this is a little bit more reasonable. But even that is a little oversimplified. So if I think I've cut out the right one, the last thing we want to do is confirm that it's the correct gene. And the way that we do that is we send it off to a sequencing lab. And what they do is they essentially take your gene sequence, they put it into a test tube with a DNA polymerase. And if you remember from replication, the job of polymerase is to come along and create a copy of our DNA. And so DNA polymerase is actually going to make a copy of that gene millions of times. And the way that we can actually kind of employ that to be able to read off a sequence is when we combine it with A's, T's, G's, and C's, which the polymerase will use, but that have fluorescent tags at the end of them. So polymerase will come along and it's going to create a copy of your original gene, but there's a certain amount, like there's a small amount, 10 or 15% of these, of the A's or of the T's or G's or C's are going to contain the fluorescently tagged ones. And so what we end up seeing is fluorescent tags get incorporated at random. The DNA polymerase just picks up an A and it may or may not have a fluorescent tag, but where that tag is added is where the sequence stops. And so these giant machines basically pretty much kind of replicate or mimic this process of replication, but with these fluorescent tags. And in so doing, we can kind of organize these fragments according to size based on these tags. And essentially, we're reading off the sequence because the C tag looks different than the A, which looks different from the G and the T. And they truly are different colored, just like this. Okay, there's purple or blue, there's green, there's red, and then there's yellow. So it's actually easy to see. You get a little printout when you do DNA sequencing that gives you your exact sequence. And so you can go through and count and make sure every one of those bases is correct before you move on and actually start to do something with your gene of interest. Okay, so again, DNA sequencing, just to repeat one more time, or gene sequencing, identifies the order of A's, T's, C's, and G's that make up a gene or any fragment of DNA. Okay, so thinking about what this means for humans, we have something called the human genome, and the human genome was mapped, okay, back about almost now 20 years ago. It's been a while. But there was a whole project involving researchers across the world that were all working at the same time to try and understand this human genome. Now, when I say it's been mapped, that means that we actually know the order of letters that make up the genes that we find on our chromosomes and the order of letters of all the other stuff in our genome that doesn't actually code for a protein. Remember, we have lots of other what we call non-coding DNA, or those introns that have to be removed during RNA processing after transcription. So there's lots of other stuff in there too. But we can tell you, if you were to look at chromosome 1, you can find the information about the order of A's, G's, C's, and T's that make up that entire chromosome down the whole length. Okay, which is pretty cool. So we've done that for all 46 chromosomes, including X and Y. But what comes next then is trying to identify what all those different letters represent, right? What is it that our genes are actually doing for us? So we think humans have about 20,000 genes, and this is a pie chart showing the functions of different genes that make up that 20,000 number, but more than a quarter of these are completely unknown. We have no idea what the genes do. We know that they're genes because we know that they'll produce a protein, but we have no idea what that protein is or what happens if it's, dysfunctional and mutated, or, so on and so forth. And see, a lot of our proteins here have to do with the production and breakdown of RNA and DNA. That's nucleic acid metabolism, cell-cell communication, so how our different tissues respond and communicate with one another. Protein metabolism and modification of our proteins are what do all the work in our cells, so the production of proteins is super important. Other metabolism, so again, breakdown of sugars and other food items, cell growth and development, so these could be those cancer-causing genes that lead to issues with the cell cycle, immunity and defense, transport, cell structure and motility, so movement of cells, protein trafficking, so meaning how we send and receive proteins from different areas. And again, here's your miscellaneous and unknown. Okay, so this is still very much an active area of research where people are trying to, assess what are these genes doing. And so the techniques I described for you here are going to be those things that people are actively using in labs like right now. Okay, and it's pretty simple at this point to do all of those because this information is so widely known and shared. So within our genome, I mentioned we have about 20,000 genes, but in reality, we have a lot of other stuff, that non-coding DNA I talked about, that we don't think does anything. So in total, we have about 3 billion bases that makes up our total of 46 chromosomes in each cell. If you think about that, that's a lot of A's, G's, C's, and T's. But among those 3 billion bases, the newest thought is that only about 2% of those 3 billion actually create a protein. Now, what the other 98% do or is doing, I have no idea, and that's something that is certainly of interest to a lot of people. But this is kind of showing you a comparison of different types of organisms and the sizes of their genomes. So again, humans has 3 billion. We're right here in green. Very similar to mice. Very similar to all sorts of different things. There's fish, there's a plant down here, there's bacteria, a lungfish up here, which is a pretty archaic kind of ancient fish. A newt has more than us. So the reason why this diagram is up here for you is just to kind of emphasize to you how complicated something is has nothing to do with how many bases are in their genome. We can have super simple organisms or simpler animals like this lungfish or a plant, right, that can have very different numbers as compared to humans. But humans are, by and large, the most complex species that we've got. And so, in thinking about this, complexity has nothing to do with the numbers of letters that we have, more so with the types of proteins we get from the genes that we actually express. And that's something, again, that we're still trying to look at. So when we think about humans specifically, I mentioned that DNA fingerprinting, we all have our own DNA fingerprints. And that comes from these slight differences in our genome. So we know that any two humans probably differ in about 1 out of every 1,200 or so bases in our 3 billion bases. So that's a significant number of differences. And remember, every time our cells are going to replicate and go through the cell cycle, we can end up with more mistakes, right? That then can be passed on to more cells. So using these technologies really help us to identify differences between individuals, identify and kind of pinpoint disorders and mutations, and so on. And a lot of this ties in with genomic testing, and there's been a lot of conversation about that here lately, especially with these like 23andMe tests where you can look at your ancestors and find relatives that you never knew you had, and so on. But the problem with some of those is there are some ethical and legal kind of considerations in that you want to make sure that that information that you're obtaining is really private to you and not to people like your employers or your insurance company and so on. And there's not a lot of regulation with that. That being said, again, this type of research is super important in helping us to identify issues with health in the medical field and again, disorders and mutations. And so understanding that comes with this type of technology, understanding our genome, understanding our differences, and so on. And so there's got to be a little bit of a balance there in that case. Okay. So that's all I've got for section 15.3. As with all of these screencasts, if you need anything, let me know. Otherwise, I will see you next time. Bye, guys.